Hello everyone, welcome back to the BGCSE past paper review. Today we're reviewing a biology paper. It is an alternative to practical paper, which is a paper 5, and this was from the year 2020. Now again, before you start the examination, always ensure that you write your school number, your candidate number, your surname, and your initials. Again, read through all the instructions carefully before you start the examination. Also, if you have any questions or any queries, please refer to your examiner. All right, let's get into the first question right here. And question number 1 is a student carried out an investigation to test a sample of food to see if it contained sugar, starch, and protein. So this is our food test. The student used the method listed below. Method number 1 is said that label 3 test tubes A, B, and C. 2, add a small sample of food to each test tube. Measure 10 centimeters cube of water. Pour into test tube A and mix well. Do the same for test tubes B and C. Number 4 is add 4 drops of iodine to the test tube A. Observe the color scene. For number 5 is add 4 drops of burette solution to test tube B. Observe the color scene. For number 6 step is to add 4 drops of bendic solution to test tube C. Using a Bunsen burner for the 7th step, use a Bunsen burner flame, gently heat test tube C, observe the color scene. Now for option A, or part A of this question, which is part A1, is to state the name of the apparatus used to measure 10 centimeters cube of water in step 3. The best instrument to use to measure 10 ml, which is a small volume of water, it's best to use a measuring cylinder. Okay, so a measuring cylinder is the best thing to use there. A measuring cylinder is also called a graduated cylinder. Okay, the measuring cylinder or a graduated cylinder. All right. Now, a beaker is not good to measure 10 ml. That's the point I thought. Okay, a beaker normally have higher gradients and the smaller markings on them is not necessarily good to use a beaker. But yeah, a measuring cylinder is the best thing. For part 2, is to state 2 things the student should do to accurately measure 10 centimeters cube of water using the apparatus in A1. So once you're using a measuring cylinder to measure volume, there are 2 things, well, more than 2 things you need to do, but 2 major things that you need to do is 1 is to use a dropper, all right, to add a few drops. So let me say it this way. Use a dropper to add the final few drops, the final few drops of water, all right, to few drop of water up to 10 centimeters cube, okay? Up to 10 centimeters cube. So again, you can pour the water into the measuring cylinder, but when it reaches close to the 10 centimeters cube, you then use a dropper to drop by drop to make sure it's really perfect. Because if you just pour it out one time, that's 10 ml, it's going to be a little bit over, a little bit less. So that's one way to make it accurate. The next thing you need to do is to read at eye level all the time. Read these instruments at eye level. Read the measurement or the volume. Let's say read the volume at eye level. Okay. All right. So you need to make sure you do that. And the reason for this is to avoid parallax. Okay. So read it at eye level to avoid, let's say, parallax. And parallax is an error. So to avoid parallax, error, all right, 2 R's, for 1 R, 2 L's, all right, parallax error, all right. All right, so yeah, so those are 2 ways you can do it to ensure that you minimize errors and increase accuracy. Not only that, what you can also do, make sure you do it on a level surface as well. So you can put the measuring cylinder on a level surface and take your measurement, all right, so eye level, level surface, important. All right, so let me just say use a level surface, because if it's on a slanted surface, you're not going to get the correct measurement at all. So use a level surface And when I say level surface, for example, a level surface could be like a tabletop, right? This could be a tabletop. Well, that is if the table is perfectly level as well, too, because that's the next problem. However, part B, I guess you get the idea. For part B, so list 2 safety rules that should be followed when using a Bunsen burner. All right, so if you use a Bunsen burner now, which sometimes we don't do, but we should. And so let me put the first one is to wear safety goggles, yeah? All right, so wear safety goggles. That's very important. So that's one. What you can also do is to use a heat mat. Let me put this one right here. Use a heat mat as well. So use a heat mat as well. Use heat mat. All right, so that's also important. All right, what you need to do as well, a safety precaution as well, you need to close the point that's called an air hole. So you're going to close the air hole on the Bunsen burner. So close the air hole on the Bunsen burner. All right, on the Bunsen burner. Before lighting. All right, so before you light it, make sure close that air hole. Then after you finish lighting the Bunsen burner, then you can open it to adjust your blue flame. All right, but before you should keep it closed. And the purpose of that is really to allow gas only to come through one part rather than going sideways. And you may have the gas flowing sideways, you light it, and boom, the lab is blown right up. All right, so to prevent that. What you can also do before you actually do the lighting process is to check for leaks. You can check for leaks. So turn it on, make sure you're in, turn off the pipe. If you're hearing leaks, do not turn on the Bunsen burner. If you turn on the gas line and the Bunsen burner is off and no leak, then yeah, you can go ahead and start using the Bunsen burner. So check for possible leaks. That's another safety thing you can do as well. Possible, let's say, gas leaks, right? Possible gas leaks, because you don't want to light a Bunsen burner in a lab that is smelly with gas. You don't know where the gas is coming from. That can be really, really dangerous. It can cost you your life. All right, so those are some of the basic things you can do as a safety precaution in using a Bunsen burner. All right, so next part now. So part C is complete the table below to show the results obtained by the student. All right, so here now we have test tube A, you have iodine, you have nutrient tested for, you have the color seen, and the nutrient present. So iodine is test for starch. Let's put starch here. All right, and we know you know that. All right, let's test for starch. And the color, if it's a blue-black color, then yeah, starch is present, yes? Burette is used to test a protein. The color seen will be purple. All right, so the color seen here will be purple. And yes, it is present, right? Benedict solution is used to test for reducing sugar. Or if you say sugar, that is also fine, but the specific one is actually reducing sugar, okay? This is test for reducing sugar. If you say sugar, yeah, you're going to get the mark. All right, orange. And if you see orange, if you see red, if you see brown, then yeah, sugar is present. Question 1 is finished. Just like that. Question number 2. All right, I put this in this kind of shade so we can see the pictures clearly. If it was darker than this, then you won't see it so properly, right? So you notice the shade of the background kind of change a little bit. All right, so for question number 2 now, so a student collected 3 different flowers from 3 different plants in the schoolyard. You have flower A, B, and C, yeah? Now for part A, it's using the photographs of the flowers shown above, complete the table below to compare these 3 flowers. All right, let's talk about these 3 flowers. All right, so first thing here, you have the Bohemia, which is the poor man's orchid. So it's the poor man's orchid. And we have the hibiscus plant with the morning glory. And I could tell you which one is which one. They're not in order up here, you know. So you have to know. They should have given order. But it's not in order. Because A is hibiscus. B is there's a poor man's orchid. And C is the morning glory, right? And most of us will know what hibiscus look like, but not everybody actually know what the Bohemia plants look like or the poor man's orchid look like, right? Yeah, but that's what it is. It's orchid, if you notice some other fact. So if you look at the shape of the flower or the design of the flower, you know that's the arcade. Indeed. All right. So uh here now we said we have four columns really. We have the name of a flower, we have a flower name going across like that, and then we have number of petals in the second row, and then the third row we have our stamen observed, yes or no. So whether or not you have stamen or not. All right. The fact of the matter is the Bionia plant here, the number of petals in this is only 4. And based on this picture, 1, 2, 3, 4. So 4 of them, right? The hibiscus plant is the first one. If you count the number of petals, we have actually, I could actually take that, yes. So 1 there, 2, 3, 4, 5. So we have 5 in that one, and let's just show you the 4 I'm talking about here. That's 1, 2, 3, 4. Okay, so the pores market has 4, and the hibiscus has 5 petals, right? All right, so just to do this, you get 2 marks there. That's a 3-mark question, right? And here now it says stamen observed. If it has a flower, it must have stamen, isn't it? Yes, a stamen is present, because remember it's a flowering plant, isn't it? Yeah, so we should be good at that. So I'll say, yeah, once a flowering plant is a flower, flower has 2 parts. Well, you see, let me correct that still, because the truth is though, if you look at the corn, the corn, the female flower is different from the male flower. For which the male flower is called the inflorescence on top, and the female flower for the corn is coming through the corn itself, or the corn and the cob itself, right? So they're actually in different places. So let me just state about that a little bit. But generally though, flowers do have the stamen, all right, and the pistil, which is similar as the carpel within side of the flower, all right. But very few of them will have them separately, all right. Such as the corn. Corn is a common one. All right. Part B is a state one other observable difference between these flowers. So look at the flowers carefully. But what was the other? So they're talking about the number of petals, right? So not the number of petals, not the not if you see stamen. So besides stamen and besides stamen present, and also the number of petals, what other things are different between these flowers? So look at them carefully. There's a number of different things that you can actually write here, though, if you think about it, right? The design or shape of the petals is one. We cannot talk about the color because I don't know what the color is, right? But of course, the design of the flower is different. So one we can talk about is the position of the stamen. The stamen, they are positioned differently. Notice the stamen here on the first one. You're not going to see clearly on this diagram, but the stamen, they are close to the stigma for the hibiscus. The pores monarchid, notice the stamen, they're actually hanging away from the stigma, so they actually hang outside of the petal. And for the morning glory, the stamen will be inside of the flower, right? So the position of the stamen is what is the biggest difference right there. And of course, how close the pistil, the petals are. For morning glory, the petals are really, really, really close. They are joined together technically almost, or overlap. Let's say overlap, they're overlapping really close. The pores monarchid, the petals are really far apart. And for the hibiscus, the petals are slightly separated, right? Are slightly apart. All right, so yeah, so the closeness of the petals could be one, or we can have the position of the stamen, and also the number of stamen. So we have 3 things to talk about. So one, we can say the position of the stamen, position of the stamen. That's one. The position of the stamen, that could be one, or we can put another one here. I'll talk about is the distance of the petals, or the closeness of the petals. So is the distance or closeness of petals. Distance of petals from each other. All right. So or close they are. And I also talk about one as well. There's other one we talk about is, which one I talk about again? That's like position of a stamen, the distance of petals from the thing. There's one where I actually saw, the number of petals, the number of stamen, yeah. Yeah, because the stamen will be different. The number of stamen will be way different in these flowers as well. The Purban's orchid has less than the hibiscus. Hibiscus has a lot of stamen. The diagram is not so clear for you to see it. And also the number of stamen within the morning glory is also different compared to the two. So the number of stamen could also work as another difference as well. They must put number of stamen, all right? Number of stamen. All right, and stamen is the male part of the flower, just to point that out, all right? Now, part C. And part C1 is to measure the length and width of leaf B, use a suitable unit. I'm going to show you on the diagram what actually was measured. And so just in case you have something to measure, talk about. Well, let me just show you where the leaf is, as a matter of fact. So this is the leaf. It's a kind of a funny design leaf. If you notice how it open, I'm just tracing around the leaf. Okay, and it gone back to the floor. So the leaf is like, that's the entire leaf right there. All right, that's the petiole. And we measure across it to measure the widest part right there. That's across. And you're going to measure the length, which is the longest point right here as well, right? So the longest point in both directions, that's what you're actually going to measure. And so I've done this measurement already. And so I have my values. And so the values here, what I got for the length was 1.8. I'm going to point this out again. 1.8, you can measure in centimeters, or you can measure in millimeters. 1.8 or 18 millimeters, right? So just make a note of that. All right, for small measurements, millimeters are the best, because millimeters will give you those small, fine readings. All right. And the width is 2.1 centimeters or 21 millimeters. All right, so those are the possible measurements right there. All right, it's a year right here. So draw a diagram of leaf B twice the size in the photograph. So I want to point out something really quick now. To do this, because this is a magnification, they actually test it on. They test it on magnification. So let's talk about how you're going to do this to make this accurate. One, you take a ruler and you measure the width across like that, right? And to measure the width across, what you're going to do is to measure twice as much as 1.8, because 1.8, sorry, the length is 1.8. All right, so you're going to measure twice as much 1.8 and going across. So 1.8 times 2 will give you 3.6. You're going to measure 3.6 centimeters across. So what I will do is put my ruler here and I measure from here to here, and that must be 3.6. Sorry, that's the width. 3.6 is the length. That's a mistake right there. So let's take that off. Let's do the width first. The width will be 4.2, right? Centimeters. So you go 4.2 centimeters across here. So your leaf must be in those position. And then the length now, that will be over 3.6, starting from here to about here, right? So I want to make the highest point here. So you just do some small dots to kind of guide you. And so remember the leaf shape again, you're going to come all the way from the bottom here. Make sure you follow through here with the side of the leaf. And the highest point going to come there. Then you can always curve like that. And then you come back this side here to make the side of the leaf touch that point. And you could come in here now and you draw it like that. All right, so that's an easy way to kind of determine where your diagram will fall once you have the magnification, right? So the magnification is times 2. So once you have done that, you can always put the extra stuff in as well. You put your leaf details in. Look at the leaf, you put the details in. So you do something like that. All right, so let's put a few of these there. All right, you can also put the extra details once you're drawing to make sure you have it almost accurate. Now, when you're drawing this in a lab, you don't need to put every single detail, like all the leaf veins, but yes, you need to put that as close as possible. The important thing is you want to make sure the outline is actually accurate, and they can put the details in afterwards. All right, so leaf will look something like that. All right, I'm not that artistic at all, but yeah. All right, on the diagram now, it's on the diagram of the leaf drawn in C2, label the vein and petiole. So let me just put a line right here to represent the vein and the petiole. So let me just do them now, label. So the first one I drew, that was the petiole. So the petiole is like the leaf stem. So that's the petiole, that's the leaf stem. And then this will be the leaf vein, or I just say vein based on what they have there. So this is the leaf vein there. All right, so that's that for that question. The question, oh wow, the question actually finished. Yeah, that's a total 10 marks at the bottom there. All right, let's go to question number 3. I think we're moving really good. I like that. I like it. I like it. All right, so question number 3. So the following question is linked with studies on vitamin C. All right, it's a student investigating the vitamin C content of several liquids by measuring how much liquid was needed to decolorize potassium permanganate. The students repeat the experiment 3 times. The data collected is shown in the chart. All right, what I want to point out here, right? There are more than one ways. There's more than one ways to test for vitamin C content in a liquid. All right, this is a basic one because vitamin C is acidic, right? And so if vitamin C is acidic, then it will decolorize the potassium permanganate because potassium permanganate decolorize in acid. So more drops means that acid is weaker or less. You have less amount of acid. Few drops mean that you have a lot of acid, yeah? Okay, that's the point that out. In the juice, you have a lot of acid, then you have few drops. If a liquid contains little to no acid, then it requires much more drops. All right? Now, there's another way to test for vitamin C. I think you need to know it. Is I use a special solution called DC-PIP, okay? So it's called DC-PIP. So DC-PIP is another solution I use to test for vitamin C. It's a dark blue solution, and this is called dichlorophenyl indophenyl. All right, so I'm gonna just write the name for you right here. So it is dichloro, okay, phenyl, all right, phenyl, indophenyl, all right. A matter of fact, to be specific, is actually 2,6. Let me write the next part right here. There's actually DC-PIP is actually 2,6. 2,6 like that. All right, dichlorophenyl indophenyl. All right, so that's the name of the thing. DC-PIP is also acceptable because they do not always expect you to know the name of it. All right, but I'm saying just in case you see to know that is used to test for vitamin C. All right, so here now, it's just to investigate vitamin C, and we talk about that, okay, 3 times, blah, blah, blah. This is the data in the table, and let's investigate the data. All right, now you have the liquid tested. You have grapefruit juice, you have pineapple juice, you have orange juice, you have orange drink. Okay, those are 4 liquids. And the different tests, experiment 1, experiment 2, experiment 3, and so they have done this 3 times for each of the juice, and then they get the results here. Now, there's a calculate the missing average value for orange juice. Write your answer in the table. So the average here is 1 plus 2 plus 3 divided by 3. And we get here is 2 because it's going to be 6 over 3, which is 2. So 2 is your missing value. That's the average number there for orange juice. OJ is 2 drops. All right. Let's go to the graph now. So draw a bar chart of the average data for each liquid tested. So I'm going to skip through this and show you what the diagram look like or the graph look like. So the graph will be something like this. Scaling is important. A matter of fact, always label your axis. Please label each axis and also make sure you use appropriate units as well. So we have number of drops on the Y, the liquids tested on the X, and so these are the bar graph right here. So grapefruit and pineapple, they have the same average. Orange juice only only required 2 drops to change potassium permanganate, and the orange drink requires a lot more drop, which was 11. And what you need to do as well is I shade these, I use different patterns. So what you can do is to use a key. So you can have a key on the side of the thing, just to outline that as well. So if we use patterns like what I did, please ensure that you have a key to indicate what is what. Even though it's straightforward from the bottom, it tells you what is in what, but a key is important if you're going to use different patterns, right? All right, so let's get into the question now. It said, which liquid contained the most vitamin C? Explain your choice by referring to the graph. All right, if we go back to the graph, you notice that orange juice requires the least amount of drops. So the least amount of drops, which is 2 drops, to change potassium permanganate. So orange juice, OJ, is the answer right there, right? So orange juice is our answer for this question. Let's write it down. All right, so orange juice. All right, and orange juice, the reason for that, because I explain the answer. So it uses the least number of drops. All right, it uh, uses uses the least number of drops, all right, number of drops to change the or to so change or let's say decolorize, to change or decolorize, because that's exactly what was happening. So the change or to decolorize potassium permanganate, okay? All right, so decolorize potassium permanganate. I put that O right there. All right, to decolorize potassium permanganate. All right. All right, so use to decolorize it. Potassium permanganate, let's say potassium permanganate. All right. All right. Oh. Right. Yeah, good. All right. So use it to decolorize potassium permanganate. All right. All right. So that's that for that part of the question. Now let's go to the next part, which is part 4. It says suggest why the students calculated the average. Why do you calculate an average is to increase accuracy. That's simply it. All right. To increase accuracy. All right, or to obtain a more accurate result. So, so let's say for more accuracy, or for more accurate estimate, because your estimation, right? More accurate estimate of the number of drops. All right, required. All right, so yeah. So it's really for accuracy, right? So accuracy is the key thing. It's to really to increase accuracy. All right, that's the thing right there. For part D, which is final part of this question, it said the same investigation was carried out on the same orange juices a day later, and vitamin C content was found to be less. Explain why. All right, so vitamin C, just make a note right here. Vitamin C deplete over time. That's the first thing you need to know. All right, so vitamin C deplete over time. All right, so that's something that we need to know right there. All right, so the next thing now, if the juice was not stored in a cool place, for example, right? So let's put that down. So if the juices were not stored, all right, were not stored in a cool place, I know a cool place or area, the heat, could have, let's say, break down. Let's say break down. Or let's use the word degrade. To break down or degrade the vitamin C. All right. Yeah. So it could break it down or degrade it. Okay. So break down or degrade the vitamin C content. All right. Because of course, because of heat, it will react because of chemical substance and then eventually to the breakdown. All right. So definitely that's why after a while juices are not even good because the content of it actually break down or react based on spoilage or fermentation. Now, number 4, again, because of the pictures and better able to view them, then you have a little bit of a darker background here. All right, so that's the best I could do. Now, it said now biological detergents are said to be better at working, at removing stains from clothing than non-biological detergents. A student gathered the following apparatus to test this hypothesis. And here now you have cloth stain to stains. You have a cloth to stain. Okay, that's important. The cloth to stain. We have 2 beakers. We have a stopwatch, and then we have blue dye. So the blue dye is what we're going to use to stain the cloth. All right, and we have a water bottle with water in it, and then we have here detergents, and we have 2 detergents here. They are Cyclone. One is non-biological, and next one is biological. If you could see it on the thing, right? Let me highlight it for you. So one package is the non-biological. The other one is biological. And you have a glass rod. So here now it asks us to Use the material listed above, write a suitable method to show how you can determine which detergent is most effective in removing stains. All right, and if you notice, as I mentioned earlier, that typically you get a lab method to write out in one of the in paper 5, right? So the first thing you do is you have to label each beaker. So let's say label each beaker. So let's say number 1, you're gonna label each beaker. All right, that's very important. You're gonna label each beaker, and you're gonna label them according to the detergent you're using. So you're gonna say, okay, non-biological and biological. All right, so you label them. You can actually say 1 and 2 and know what 1 and 2 is. All right. Next thing we'll do is to place the cloth in the beakers. We're gonna place one of the cloth in each beaker. Okay, that's very important. And next thing we're gonna do now is to, after do that, place the cloth in each beaker. Let me just put another point right here. You're going to pour some water over it. So you're going to pour water over the cloth. Pour water. I have too much O's right there. So pour water in the beaker. Cover the cloth. All right. A matter of fact, let me just point this out. I'm going to use the same amount of water. Pour the same volume. Let's say this. Pour the same volume. You want to make sure you make everything be constant, right? Pour the same volume of water in the beakers to cover the cloth. All right. Yeah, that's very important to make sure you use the same volume. And next thing you can do right here with the 4th step. After you do that, you're gonna pour equal amount of dyes. No, so you're gonna pour equal amount of dye. For equal amount of dye in each beaker. Okay. All right, and you notice I pour water first. You don't have to do that. Actually, what you could do, you could put a drop or 2 drops of the dye in a specific point on the cloth directly. So you make that part alone be stained. So you stain that part alone. So what you can use is the method I'm doing now is to stain the entire cloth. And why I'm doing that, because if I use a drop or 2, I don't know how far the dye may spread, but I want to stain the entire cloth. All right, that's what I want to do, stain the entire cloth. But you can also do it and stain only a portion of the cloth. All right, so you can also do that. But since this cloth, the dye is going to spread a little bit, right? So you can even draw like a small circle onto the cloth and put the dye there alone. You can also do that. But again, I am saying that it may spread a little bit. Or even for better yet, you can use the same number of drops of dye in a specific area on the cloth. So whether it be the center part of the cloth, which will be the best part, and you can do it like that, right? But I want my entire cloth to be covered with dye. All right, so the 5th thing I want to do now is to add different, or add the different, let's say add the different detergents in the specific beaker. All right, and why? Because when we already label them, you say, okay, one is the biological, one is the non-biological. So you put the detergents in the specific beaker, or their respective beaker, in their respective beaker. Let's say respective beaker. All right, rather than say specific. All right. All right, so let's say respective in their respective beakers. All right. So once you have done that now, the next thing you want to do, which is our next point, 6th point. After you do that, you're going to let the beaker sit for a specific time. Let the beakers sit for before I even start do that, I need to stir. Because when I want to put the detergent in, you're going to stir it a little bit to make sure the detergent work its way through, right? So you're going to use the glass rod. Use the glass rods to stir the detergent in the beaker. And you want to do that, I want to make sure you use equal amount of force. Do not spin one harder than next one, because that could also create a disadvantage for one or an advantage for another. The next thing you want to do after you do that now is to let the beaker sit for a while. So let's put this in number 6, a matter of fact. Let the beakers sit for a specific time. I sit for a specific, all right, for a specific time. That's very important. Must be a specific time, all right? Just put that piece there. And our 7th step now is the last step. You're going to remove and rinse. Okay, you want to make sure you rinse off the excess thing. Or if that is loose, then that you're going to rinse off very easily. So remove and rinse each piece of cloth. Okay, each piece of cloth. All right, so you're going to make sure you do that. The cloth must be the same size as well, just to mention that as well. So you're going to remove and rinse each cloth. And then observe for any difference. Okay, so observe for changes. Yeah, so pretty much that's what you're going to do right there. That's 7 steps. All right, so cool. All right, and that's for 6 marks. 7 steps for 6 marks, so we're good. For the B part of this question now, is to make the investigation fair. What 2 things that must be controlled? And there are a number of things you can control here. You can use the same amount of dye. And I think I mentioned that in the steps. So we can use the same amount of dye. Use the same amount of dye. You need also the same amount of dye. You can also use the same amount of water. So use the same amount of water. Use the same volume of water. All right, you're going to do that. And next thing you can do to ensure fairness is to soak the cloth for the same time, or the same amount of time. Soak the cloth. For the same amount of time. All right. Yeah. So make sure you have the same amount of time for each cloth. Next thing you can also do again is to ensure the same temperature of water. Yep. So use the same temperature of water, the same type of water. So the same type and temperature. So there are a number of things can the same type or temperature of water, because if one water is hard, when I mean hard, I mean contain minerals, then the detergent will not lather properly and will not take out the stain easily. And if the water is soft, and both of them are soft water, which come from the same source, then yeah, you're going to work pretty much equal. So the water will not affect the effectiveness of the detergent. The temperature also could also damage, if the water is too warm or too hot, then it will damage the enzymes that are placed in the biological detergent, right? And so that will also cause you to have errors as well. So the water temperature used must be constant and must be at a moderate temperature as well, not too hot, not too cold, right? You want it to be fair. Give the enzymes time to work. All right, next part of the question here, which is the last part of this question. It said, what are the expected results from this investigation? Give a reason for your choice. And so the expected result is that the biological detergent will remove most of the stain. All right, that's just it. All right, the biological detergent will remove most of the stain. And again, we're just saying this because that's what we expect to happen, right? And notice that the expected result to remove most of the stain. And the reason for that is, I give a reason for that because the detergent, which is biological detergent, so the biological detergent contains enzymes, all right, that will break down the stain. Okay, that will break down the stain. All right, and that ends this paper review here. Again, good luck, and I'll see you soon. I'm going to see you plenty of time. So keep safe, keep in tune. All right, talk to you later.